I'm George Malliaras. I'm a professor in the Department of Engineering, and I'm an experimentalist, which means I should be able to load my slides, right? You would think that. Yes, here it is. Excellent. Can you see my slides? Great. So I'll talk about flexible electronics on the brain, very general type of presentation, which is motivated by the fact that neurological disorders are the leading cause of disability worldwide, with many million years lost to stroke, migraines, dementias, epilepsies, and so on and so forth. There is good news that comes from the field of bioelectronic medicine. Powerful demonstrations such as the one shown here on the video, where implanted electrodes are used to stimulate the brain or the spinal cord or peripheral nerves and achieve an amazing effect with just a flick of a switch. This has been used in motor disorders, has been used in epilepsy to stop seizures, in neuropsychiatric disorders, in conditions such as obesity, and many more. So very exciting field. What prevents it from being deployed to scale? And there are some serious limitations that we need to address. First and foremost is we do not understand how the brain works. It's a very complex machine. And despite the fact that we understand how neurons communicate with each other on a cellular level, we do not understand how we go from that to macroscopic behavior. In order to address this question, electrophysiologists use electrodes at different configurations, implanted on the cortex, on the scalp, to try to listen into neural communication and correlate it with behavior. The opposite is also not understood, how you go from stimulation to changes in behavior, the so-called mechanism of neuromodulation. Now, this lack of fundamental understanding goes hand in hand with technology limitations. The signals that are detected here in the brain are very small and diverse, can be small metal ions, can be larger molecules, can even be components of cells being exchanged. The environment is hostile to electronics. When you put a probe in the brain, is bad not only for the brain, but also for the probe. And often the surgery that's associated with the placement of bioelectronic medicine products is quite massive and invasive. So we need to address these technological limitations to be able to learn more about the brain and enable bioelectronic medicine. If I'm to plot, to discuss this in a bigger picture, paint with a broader brush, I would say what we try to do is to make electronics compatible with biology. And there are mechanical differences. You have soft versus hard. There are communication differences. You have complex signals here, simple electronic currents in the world of electronics. You have dynamic phenomena in biology, while electronics is static as we know it. So today we will hear a lot of beautiful talks about addressing the mechanical properties mismatch. I would like to focus on the communication gap and then addressing the dynamic versus static nature. So one observation here is that current electrodes are very large, and here is an example of electrodes that are placed in the brain, implantables. You're talking about millimeter size electrodes. Same for cortical arrays. Again, the electrodes here are of the order of a few millimeters in diameter. And wearable electrodes are even larger. You go to centimeter scale. And the reason for that is that the signals are weak and you need to integrate over a large area, which means you lose in terms of spatial resolution. In order to address this, we use materials such as the one shown on the right-hand side. It's a conducting polymer that can uptake ions from a solution, whether this is the cerebrospinal fluid in the brain or sweat at the surface of the body, they can uptake these ions and couple them with electronic charges in the volume of the film. Contrast this with the electrodes that I showed you in the previous slide, where ionics and electronics just interface across a two-dimensional interface, a surface, and as a result, their coupling is weak. So by going to this concept, by using materials such as the conducting polymer shown here, you can have much stronger coupling, which means you can make small devices the size of the soma of a neuron, tens of microns, integrate them with flexible surfaces, and then get really high resolution recordings. You can also build additional functionalities. For example, you can make those optically transparent and use them in conjunction with super resolution microscopy to get really a very fine level of detail. Here we're looking at proteins expressed in the synapse of two neurons. And you can also make them MRI transparent. And this technology has been used to achieve ultra high resolution electrocorticography down to the level of a single neuron, as well as measure very high resolution activity from muscle from the surface of the body. So here you have to go to millimeter scale because we are talking about being far away from the source of signal, and the signal is very weak. But still, millimeter means that you can think about very articulate myoelectric prosthesis now. Another topic that I would like to very quickly discuss is the dynamic versus static nature of biology and electronics. And this translates into, this I can put in the context of minimizing the invasiveness of surgery. So here's the placement of a cortical grid. It requires a large craniotomy. The placement of a paddle electrode requires a laminectomy. This is in the spinal cord for a relief of pain. And looking in the field of soft robotics, you see a lot of examples of objects changing shape as this soft robot demonstrates here through pneumatic actuation. So the idea is, can you use these concepts in bioelectronics? And sorry, here is a cortical array that has microfluidics, and you can use this microfluidics to change the shape of the array. So by doing this, we were able to make paddle electrodes and then roll them into a small needle-shaped electrode that can be implanted in a minimally invasive manner and then deployed inside the body. This was demonstrated in an in vitro model, but also demonstrated in a human cadaveric model that maintains the correct anatomy. Very exciting news because it points to a future where you can envisage doing keyhole surgery for placement of neural electronic devices. So with that, I would like to summarize. I talked about bioelectronic medicine as an exciting new field in the fight against disease. We talked about mixed conductors for better electrical communication, better electrodes, and microfluidics embedded in these implants to minimize invasiveness of neural surgery. Let me thank my collaborators, funding agencies, and thank you for your attention. I will now look if there are any, to see if there are any questions. I see no questions. Is there anyone who would like to ask a quick question?
Hi, George.
Yes.
Thanks a lot for your great presentation. You highlighted several technology. I wonder, most of them were actually invasive, but I had the impression that there were some non-invasive that can be put on the surface of the scalp or skin, correct?
Correct, yes. So the skin is a good target because obviously it's non-invasive, and surprisingly enough, you can do quite a lot of interfacing with electrically active organs.
And how far we are in the technology? How mature is this technology that you mentioned for let's say, for example, the micro electrodes that can be put on the high resolution electromyography, for example?
So there are a lot of demonstrations, and I'm sure we will see a few more examples today, including technologies that are used to in the stump of an amputated arm to drive a myoelectric line as a whole, very quickly to get us back on time.